Thanks for joining us. My name is Loren Wold from The Ohio State University, and I'm really excited to be here today with Dr. Weeks.
Hi, I'm Kate Weeks. I'm a group leader at the University of Melbourne, and it's really exciting to be here at BCVS.
Dr. Weeks, why don't you tell us a little bit about the work you're going to be presenting here at BCVS?
So, my research focuses on a phosphatase called PP2A. What a lot of people don't realize is there are multiple species of PP2A. There could be as many as 30 different types of PP2A in the heart. And these proteins localize to different parts of the cell and potentially have a whole heap of different substrates. So, our research has focused on two different PP2A species, B55 alpha and B56 alpha, and trying to understand the roles of these proteins in the heart and specifically how they regulate the cardiac response to beta adrenergic receptor signaling.
That's great. So you've obviously been working in this area for a while, and what are some surprising results that you found that you really didn't expect going in?
So, I think one of the paradigms in the field is that different signaling stimuli can activate different pools of PP2A to serve different functions. So, in the context of beta adrenergic receptor signaling, we actually see activation of multiple pools of PP2A. So, it's not necessarily switching from one to the other, it's actually activating several pools to serve different functions. So, that to me was surprising and something that we're still following up.
So are there drug targets that you're looking at?
So it's a really exciting time to be in phosphatase research because phosphatases are now druggable. We now have these small molecules, new classes of small molecules that can selectively activate or inhibit different species of phosphatases. And that means they're druggable. In the past, we didn't think this was the case. Phosphatases were thought of as housekeeper enzymes that were non-specific and constitutively active, but actually they're highly regulated. And with these new compounds, we now have the possibility of actually targeting their activity.
So one thing that's always interested me with cardiac physiology is that male physiology and female physiology can be very different. And so I'm curious, in phosphatase research, are males and females responding differently?
So it hasn't really been investigated, and there are hundreds of phosphatases that are in the heart, so it might be different for each phosphatase. But our research has found sex-specific roles for B55 alpha in the heart. So if you knock out this protein in the mouse heart, then it has a different effect on gene expression. And in a setting of chronic beta adrenergic receptor stimulation as well, we see differences in how the male and female mice respond. So absolutely there is a sex effect there, but how important that is and whether or not that would allow us to develop therapies specifically for men or for women, we need to do more work.
That's great. So where do we go from here? What's the future in phosphatase research?
I think there's so much still to understand about how PP2A and phosphatases in general are regulated. With these two species that I've been looking at, we don't understand how they're actually activated downstream of beta adrenergic receptors. We know they're downstream of PKA, but they don't seem to be phosphoregulated by PKA. So figuring out those upstream signals and the post-translational modifications that actually activate these enzymes is something that I'm very interested in. We also don't know which proteins these phosphatases actually target to modulate their cardiac effects. So we've identified some potential substrates, but now we need to validate those and understand what their function is.
Great. What's been your favorite part of the meeting so far?
Oh, well, I've just come out of one of the sessions where Zoltarani presented some work trying to understand the mechanisms of SGLT2 inhibitors, and it was a very compelling story. I think this is a question that the field has been grappling with ever since the phase 3 trials have shown benefit in cardiac patients with these drugs. So that was really exciting.
So, Dr. Weeks, thanks so much for joining us today. This is really exciting work, and I'm really excited to see where the field goes. If there's something you could leave us with to think about phosphatase research, what would that be?
I would say don't forget about phosphatases. We always think about the kinases, but phosphatases are just as important for regulating phosphorylation in the cells. So don't forget about them in your research.
Thank you.